overview of electromagnetic spectrum, just to make sure we're all on the same page. And we're going to look at absorption and what's actually happening at the molecular scale. Like, what is it that gives rise to these absorption spectra that we're very familiar with? And then I'm going to talk about some of the major absorbers in the ocean and how we characterize them and how we begin to think about absorption on sort of molecular or individual particle scales. And then what does that mean when we talk about bulk absorption, like what we measure in the lab? And this will be sort of more on just, not a theoretical base, but more of a conceptual base. And then the second lecture today, I'm going to talk about how we measure absorption. And specifically, I'm going to talk about spectrophotometry. And that's going to be benchtop, like a benchtop spectrophotometer that you might have used in the lab. And also with the AC meters, so that we can go this afternoon. We're going to go in and start working with spectrophotometers and AC meters. So we just want to talk a little bit about the theory and how they were designed, and does it really, we have a theoretical, like this is how absorption should be measured, and how close are we, because we can't do it perfectly. So what are the assumptions that go into that? And what do you have to be aware of when you're interpreting the data? And then today we'll do in the lab absorption by solutions. And so no particles in that example that I showed yesterday, no particles. This is dissolved. Meg's not here to cringe when I say dissolved, she's a chemist. And then tomorrow we'll actually do absorption by particles. And so then you'll have a good sense of that, okay? Ready for the days? Okay, all right, so I'm gonna talk about electromagnetic radiation. So charged particles, these dipoles, right? They create electric fields. And that just means that there's an oscillation between the positive and the negative. Right? So we can look at it this way, where you have these oscillating electric fields. And then when that charged particle moves, that's what creates a magnetic field, right? And so that magnetic field, which in this case is in blue, you can see they're occurring in different planes. And if you remember that when the fields oscillate, the energy propagates. And that if you take your hand, the right hand rule, you remember the right hand rule? You take your hand, you put your fingers in the direction of the electric field, which in this case, if I look at this part here, and then I curl it into the magnetic field, my thumb is in the direction of propagation, right? Everyone want to do that? Try doing it with this one, because this one's upside down. So that one, you have to go this way and curve in. We're still going the same direction, right? Everyone got that? I don't know if you ever did this in your physics classes, and you'd be on an exam, everyone's sitting at their desk and we're doing like 10, right? Okay, so, when we look at the frequencies of these oscillations of the electromagnetic field, that's what's described by the spectrum, the EN spectrum. And so we can go from gamma rays to radio waves. We're going increasing wavelength, that's what this is meant to show. And it turns out the energy goes the opposite way. Shorter wavelengths have higher energy, higher frequencies, shorter wavelengths have higher energy. And the visible spectrum, about which this class is mostly concerned, is just this really, really small part tucked in here between ultraviolet and infrared. Okay, so, yep, energy, oh, God, I always say that. Okay, so what's absorption? So since this energy is propagating, when a molecule is absorbing radiation, it's just absorbing the energy that's associated with that propagating wave. And the energy in that spectrum, and if we just want to talk about the visible spectrum, like say from blue to red, the energy associated with each part of that spectrum can be described by this equation, where the energy is related to, well, the product of the speed of light and Planck's constant divided by wavelength. And so what that tells you is, since energy is proportional to one over wavelength, that means that the blue end of the spectrum has higher energy than the red end of the spectrum. Right? Okay, and we talk, and that makes sense when we think about the black body radiation that we were talking about yesterday. Bluer things are hotter than redder things. Right? So when the molecule absorbs energy of whatever wavelength and absorbs that amount of energy, different things can happen to it, to the molecule. Okay. And so here's microwave, here's gamma rays, here's our little visible. So this is part. So here's what happens. If the molecule absorbs energy in the microwave band of radiation, there's a low and lower energy there, so it's visible. And what that does to the molecule is it makes it rotate. Right? The molecule just starts to rotate around. And the molecule rotates around. What happens to them? They've got kinetic energy, right? So they've got kinetic energy. They're going to warm up. That's why microwaves work. They're just causing rotation. So if you move into the red part of the spectrum, which is a little bit higher energy, so now molecules are not just rotating, now they're vibrating. So in this example, there's a water molecule, it can be vibrating this way, the bonds between the oxygen and the hydrogens, this way, this way. It can do a little bit of bending like this, right? So there's enough energy to cause the molecules to start to vibrate, if they can absorb that, okay? When you get into visible and ultraviolet, now what's starting to happen is you're getting what we call electronic transition. And so that's when you start moving electrons through orbitals, okay? So it takes a lot of energy to get electrons to move. And those of you who think about photosynthesis know this, because you think a lot about how electrons get moved out of reaction centers and move along the Z-scheme. It takes a ton of energy, it takes light, right? It takes that much energy to do that kind of molecular transition. Okay. If you start getting into X-rays, that's when you get electrons leaving. It's called photoionization, and it's just an injection of electrons. It takes a lot of energy to get an electron away from the molecule. But it turns out that X-rays can do that. And then finally, gamma rays, they actually change the configuration of the nucleus. Or, I'm sorry, the nuclear configuration. So depending upon where you are, different things are happening to the molecule. But we're in this region here. And so really what we're going to be concerned with are these electronic transitions and molecular vibration. Okay. So, all right. So the energy that it takes to do these electronic state transitions, that energy is quantized. And so if you're going to move an electron to a higher orbital, it's going to, depending upon the molecular structure, depending upon the orbital shapes, the electrons, et cetera, the amount of energy that it's going to take to move that electron, there's only a specific amount of energy that will do that. What do you mean by quantized? You can't have like 10 units of energy if it needs 11. 12 is too much, 9 is not enough, you need 10, right? So you've got to have exactly that amount of energy to move that electron for that molecule in that state. And so we describe this by this diagram where this is the amount of energy, and say this is the ground state of the molecule. This would be its second electronic energy level and its third. So in this case, this particular molecule, because I'm building up chlorophyll, which happens to have two states, that it would take this amount of energy to get an electron to the next electronic state, or alternatively for this molecule, there's a second state where if you gave it a little bit more energy, you could get up here. Now, if you gave it this much energy here, nothing would happen. So it can only absorb energy, the energy equivalent to get it to that electronic state. So, for example, okay. This is what determines where your absorption peak wavelengths are. So, I can figure this is really in my way. So, there's a certain amount of energy here. We can figure out, by knowing how much energy that is, we can figure out what the wavelength is. Right? We can just do that calculation. And similarly, we can figure out what that wavelength is. They're color coded for obvious reasons, right? Like, this is lower energy, it's red. This is higher energy, it's blue. So if I take this diagram and I say, okay, I have a molecule that can somehow absorb a red photon and somehow absorb a blue photon. It has that capacity to absorb that quantized energy. I can turn this on its side and say, well, that's equivalent to energy at, say, 676 nanometers. And this amount of energy to go to this third energy level, that's equivalent to, say, 440 nanometers. Okay. So that would say that this particular molecule, chlorophyll, has the ability to absorb energy in two quantized states, two different energy levels. And if we had this molecule in a solvent, and it had its molecular structures that if we had it cold, so there was no vibrational level, there was no any other energy associated with this molecule, and you measured the absorption coefficient, what you would see is two very distinct peaks associated with those quantized energy levels, specifically going from the ground state of the molecule to, say, the first electronic state, and the ground state to the second electronic state. Okay. Well, we know that in the environment that's not really how it occurs. So, so this has the two absorption peaks. Okay, so there's also, we have quantization of vibrational states, because in reality, we're not operating at absolute zero. There's some kinetic energy in there. These molecules have slight variations in their molecular structure and their orbital structure based upon what vibrational state they're in. Like if they're cold, if they're warm, right? So there's going to be different vibrational levels. Well, that means that within each electronic state, there are other amplified levels that the molecule can be at. It can be in its ground state, right? But if it's has a little bit more vibrational energy, it could be up here, or it could be here, or here, or here. And the same thing, it could be in its second electronic state with a variety of vibrational levels. So what that means is we started off with this peak, and we started off with this peak. But let's just look at this one first. Let's say one of the molecules is in its ground state, but it absorbs energy quantized up to this vibrational level. It's got some vibrational energy, right? So it could actually absorb this much energy. So what would that do to the spectrum? You have your main peak here, the red absorption, which is associated with this transition. But what if it transitioned to here?
So
Yeah. So it's not exactly broadening because it's quantized.
So it would add another absorption.
It's going to add another bar. Near this one, it's still associated sort of in that region of the spectrum. Is it higher or lower energy? This one.
Higher.
It's higher energy, right? Because it's going to the second vibrational state. It's a little bit higher energy. If it's higher energy, which side of this main peak does it occur?
Blue side.
Bluer, right? It's shorter wavelength because it's higher energy. What about, say, for example, this one, which in the first energy level was at a higher vibrational level to start with because the molecule had a little bit more energy to start with. Then it absorbed light and went to the ground state of the second energy level. How much energy is that relative to this one? It's less. This is less energy, right? The change in energy is less than this much energy.
So will that produce a wavelength that is longer?
Bingo.
Or will it produce the same wavelength?
It can't, because it's less energy. It's a quantized amount of energy that is less energy. So it's a longer wavelength. So it's going to show up on this side. So we go from having these two absorption peaks associated with these two electronic states, and then we may have all of these additional absorption events that are quantized to the vibrational states of the molecule. So, right, where am I? So that determines these sort of side peaks here. You get these side vibrational peaks. Sometimes we call them vibrational harmonics. And so, yes, it broadens, but it broadens in a quantized way. And so if we were to measure the spectrum, we would see peak, 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 right? So you know where I'm going to go next. If I've gone from electronic states to vibrational states, I'm going to go to rotational states. So each one of these vibrational levels has associated with it a series of quantized rotational levels, right? So, okay. So those are also quantized, specific amounts of energy that can be absorbed by a molecule. Can absorb a little bit more, a little bit less. It's within that rotational state. So that's when molecules can undergo rotation. It can go this way, it can go this way. Okay, so these are now begin to sort of smooth out between the electronic states, the vibrational states, and they sort of add in absorption, still quantized, in between, which begins to give rise to a more continuous absorption spectrum that we would see. And so, and you can imagine that if you had some absorption over here from some other, if this is not a specific molecule now, this is more like absorption by some other pigment or something in there. There might be some other amount of energy associated in there, from whatever state. Okay, so that's how we get absorption spectra. And one of the reasons this is important is that we want to understand, say for the chlorophyll molecule, why we get these two absorption peaks. Most other pigments don't have multiple peaks like this, especially spread so far apart. When we look at the absorption by water, we will see some peaks, and we want to know why those peaks occur. And because we have to measure the absorption properties of water in the ocean, because it's water, and we use pure water as our reference in our instruments, you can imagine that the temperature of the water is going to lead to some variations in these electronic states, right? And if we measure water that's 10 degrees versus water that's 20 degrees, we're going to get some spreading out here in different ways, right? And so we have to keep track of the temperature. So today, when you're measuring things, you're going to have a thermometer and you have to measure the temperature, because there will be variations in the absorption associated with the temperature of the water. And when we start talking about chlorophyll and measuring chlorophyll concentration, And we're going to do that with absorption, we're going to do it with fluorescence, and we're going to do it when it's in an extract of acetone, and we're going to do it when it's in a cell. Its absorption is going to be different because a molecule by itself in solution has a different configuration than it has when it's in buried into a thylakoid membrane in the middle of a chloroplast, in the middle of a cell with a bunch of cells in water, right? But that molecule, even when it's just embedded in the thylakoid membrane, has a different molecular structure. It's being pulled by bonds, and so its shape changes. And so if its shape changes, the amount of energy that it can absorb is going to change slightly, which is why when you measure chlorophyll in extract, it doesn't look exactly like chlorophyll in a cell. The peaks are different. And the vibrational levels are very different. You can see very distinct side peaks. So this is more of like what you might see in a cell. If you did it in an extract, you'd have very distinct peaks associated with the vibrational levels. So it's really important to think about when you're making a measurement of a compound, a molecule, or a mixture of molecules, or chloroplasts or organelles. Think about what the molecule's molecular state is to think about why the absorption that you're measuring, why you see what you get. It makes a big difference. Okay. All right. So one last thing is we sort of show these energy levels as being like, oh, we're here, and then this happens, and that's good. But I think it's really important to think about really what this means is we're looking at the amount of energy versus the internuclear separation. And if you try to push things together, it takes a lot of energy to push things together. If you pull things apart, you can disassociate molecules if you can pull them apart, but it takes a certain amount of energy to do that, right? So, or for moving electrons, you can pull them out. But within each one of these levels, these vibrational levels, this line here is really this line here. And there's stuff going on in the molecule, and there's certain ranges of energies associated with the actual structure of the molecule. And the point that I want to make here is that there's a probability of occurrence of where you're going to find the molecule in its ground state in terms of the actual structure of the molecule. And so if we're going to move an electron from this ground state to this ground state, well, we're moving vertically. And so, there's a very low probability in this particular molecule, I don't know what this is, but you have a high probability of being right in the center, which means this internuclear distance, which means that you're probably not going to end up in the first vibrational state here, you're going to end up in the second one, right? So there's all of these different probabilities for that molecule of where you're going to find the possibility of absorption. And this is what, if you think about what the highest probability is from one electronic state to the other, whichever vibrational level it's going to be in, that's what's going to give you the magnitude of absorption, right? So the more absorption occurs when you have a greater probability of that process happening. And so it's not that the blue absorption peak, because it was higher energy in chlorophyll, it's higher than the red absorption peak, right? Blue is always higher than red in chlorophyll. It's not to do with its energy. It's to do with it's a greater probability for the molecule to absorb from that ground state to the second electronic state. And so if you want to know more about this, please talk to Meg, because she's a chemist. Okay, but I just want you to get to this, to just understand that when we're measuring absorption, we really need to be thinking about the molecule and the state of the molecule. And if you throw it into acetone, it's going to be really different than if you throw it into a cell. And if you're looking at C-dome absorption and you have a mixture of tons of organic molecules, who's in there? What are they absorbing? What are they bound to? Is there chelation? Like all of these things that you learn in chemistry, all are going to have a really big impact when you put your ACS in the water, right? Okay, excellent. So here's the chlorophyll molecule. It's constructed of it's got this, it's really beautiful. I mean, okay, so each one of these little rings is called a pyrrole. So we call it a tetrapyrrole, so because there's 4 of them, they form this whole ring structure of the chlorine ring. And what I want you to notice is all of the double bonds, right? That's what's really responsible for the absorption. And in the case of chlorophyll, it's got a magnesium in the center. Interestingly, it's really, really similar to, oh my God, I just blanked. It's a heme.
Hemoglobin?
What?
Hemoglobin?
Hemoglobin, thank you. Hemoglobin does not have, it has iron in the center, but I think there's something very cool about the fact that hemoglobin and chlorophyll have a really similar structure. And then it's got this tail, the phytol tail, and this part of the molecule is what secures the chlorophyll pigment into the thylakoid membrane. And so you have the thylakoid membrane, and then this part basically sits out like this looking for sun. And it's this part that has the capacity to absorb the blue wavelength and the red wavelength. And we'll talk about fluorescence later. Okay, so this is the absorption of chlorophyll in a solvent, probably acetone or methanol or something. And so these are the primary absorption bands. Here are the secondary bands. It's smoothed out. All right, so what happens to chlorophyll? Things can happen to chlorophyll, the chlorophyll molecule, either intentionally or unintentionally, either in the lab or in nature, that change its spectrum. So the first thing that can happen is that you can heat it up, or you can acidify it. And if that happens, the magnesium gets removed and you get this structure. So when we do the chlorophyll analysis, how many people have done chlorophyll analysis in the lab where you've extracted it and measured fluorescence? And then what do? You have an extracted chlorophyll, you measure its fluorescence, what do? Anyone remember how you distinguish it from its degradation products? Add 3 drops of acid. You add 3 drops of acid. And what that does is what you're doing, you're removing the magnesium. And you're taking the chlorophyll molecule and you're making it a phytoplankton molecule, which is a degradation pigment. In nature, this happens too. When does it happen? Where might a phytoplankton with chlorophyll in its cell encounter acid, gets converted to phytoplankton when it's eaten, when it passes through the gut of a grazer, okay? So in nature, this happens and we end up with phytoplankton, and that says, oh, you've got a ton of phytoplankton, like, oh, it's probably the end of the bloom, there's been a lot of grazing. And so that's a natural process, and we can measure phytoplankton. It becomes the key to telling us something about grazing. In the lab, we use it so that we can separate chlorophyll from phytoplankton in analysis so that we can quantify chlorophyll and quantify phytoplankton. And we're going to do that on Friday. All right, but its absorption spectrum changes. I couldn't get one on the same picture, but here's the Soraya band, the Q band, and you can see that, we're going to move from, this is 440. This is now 410, so the bands shift. The bands are shifting because by removing the magnesium, you've changed the molecular structure of the molecule, which is changing its quantized energy level slightly. They shift, okay? All right. It also means that if you have an extract of chlorophyll and you're getting ready to measure it, you've got it in your hand, you're holding onto it, you're waiting for your station, you're going over the instrument you're going to be using, and you're standing there, you're warm because it's summer, maybe the sun's going to come out, right? You add heat, and now you've just made some phytoplankton. You're heating it up. So You want to protect your samples from heat and acid. Don't use, garbagey methanol or ethanol or acetone, because that could, acidify your sample, and unknowing to you, you would estimate you had more phytophyton. So, okay, let's go over to this part. This is blanching. What's blanching? You know what blanching is?
Isn't that like you cooking when you heat something and cool it right there?
Yeah. So there's a process, like if you really heat something up, you can actually remove the phytol tail, right? So if you let your samples get exposed to a lot of light, it absorbs a lot of light, it will heat up, it can blanch. That removes the tail. So now this molecule, basically cleave it off here. So now you have a molecule called chlorophyll. You find that in nature also. Its absorption spectrum also shifts. So you can see it's different from a chlorophyll, it's different from phytophyton. You can then acidify that sample and get phytophorite. So there's a lot of derivatives of chlorophyll that can be slightly changed by slightly changing the molecular structure. It doesn't take much. Okay. And that's true for a lot of pigments. So when you're working with pigments, you have to be very careful. And when you measure those pigments in the ocean in their natural state, they tell you something about what has gone on ecologically. So, okay. All right. So let's talk about the major absorbers in the ocean. Who's in the ocean? Yes.
Can I ask a question?
No. No.
So in the previous slide, the chlorophyll A spectrum.
Yes.
It looks like there's very little to no absorption around 500, but
Yes.
if you go back to the quantized
Yes.
blanching states, there was a green arrow.
Yes, because this is actually an absorption of a whole cell,
Oh, okay.
right? So there'd be other pigments in there, probably like a carotenoid in here. So it's not the best of the curves that could have been used to illustrate this.
Okay. Oh, that's fine. I just
Yeah.
chlorophyll A itself does not.
Chlorophyll A
have
itself does not absorb in the green at all.
Okay.
And that becomes really important when we think about the fluorometers that we use in the ocean and how we are going to calibrate them. Because it turns out you can't calibrate them to a pure solution of chlorophyll because they excite at a wavelength that chlorophyll does not absorb. Who would have thought? So you're actually exciting an accessory pigment that's in the cell that passes its energy to chlorophyll, and then chlorophyll fluoresces. But it's a really important point to recognize that. I'm glad you brought that up. Yes, you don't want to excite here. So most of our fluorometers excite at 470, which is right here. So they're actually very bad for chlorophyll fluorometers. They're very good phytoplankton fluorometers. Yeah. So when you calibrate a fluorometer, you can't put in chlorophyll and acetone. That would be so easy, but you can't do that. That's why we don't have absolute calibrations on any of these sensors. I'm just going to spill the beans right now. Yeah, excellent. Okay, so who are the major absorbers in the ocean? Who we got? Water, major one. What else? CDOM. And what does that mean? Who's in CDOM? Like varieties of molecules make up CDOM, right? Okay, what else? Phytoplankton, yes. Sediment, absolutely, suspended sediment. Detritus. Let's talk about detritus. What is detritus? And the reason why I want to talk about this is because the term gets thrown around just a lot. What does it mean? You look it up. Or what do you think it means? What do you think the word detritus means?
I don't know, but I have a different question. How does detritus vary from CDOM?
Thank you.
Is it dead organic matter?
So if you look up the definition of detritus, one of the definitions is dead organic matter. So chunks of stuff, chunks of cells, things that have been eaten, fecal pellets, once living organic matter, detritus. What if you're a geologist and you look up detritus? Suspended sentence. Right, so detritus has a lot of different meanings. It's one of those terms that I just call a garbage term. And it gets used in our field in an unfortunate way. We have phytoplankton and detritus. And I'm like, but okay, so where are the bacteria showing up? Are they phytoplankton? No, not unless they're photosynthetic ones. But if they're heterotrophic bacteria, they're not phytoplankton. Where does zooplankton show up? Turns out people use the term detritus to mean everything but the phytoplankton and the sedum. And it's a very, very bad term because it's not, they're not dead.
In the updates or?
Yeah, in the optics. Yeah, detrital absorption, they say. And so then it gets to this question of what's the difference between that and sedum? And I would say they're a continuation on the size spectrum. So you could have living organisms that are organic but not pigmented. They poop, which might have some detrital pigments in it. Ooh, detrital pigment. Yes, phytophyton, chlorophyll, phytophyton, but don't have living pigments in them. And that's clearly dead. The fecal pellet's dead. It gets broken down by bacteria living, bacteria not pigmented, but living, clearly not detrital. They release small particles in the degradation process. Particles, but getting smaller. Particles break down more and more. Suddenly pass the magic boundary, 0.2 microns. And instantly, if you're on one side, you're a particle, detrital particle, on the other side, you're a sedum. Yeah. So that's, yeah. So it's a sort of continuous process. And we separate it because that's how we made a measurement. We filtered something on a filter, right? And I'll tell you why the term detritus comes in. And now we have different things. Oh, now I have a particle. So we'll get there. So, but detritus, remind me to just keep talking about detritus because it's a term we have to be careful of because it includes things that are not dead. And depending upon how we do our measurements, it can also include inorganic mineral particles, sediments, which were never living. So do they get to be called dead? I don't know. I don't think so. It's kind of a philosophical problem, yeah.
So sorry, if it's not pigmented, but it is less than 0.2 microns, that's detritus.
That's sedum.
So they don't need to be pigmented. Okay.
And when we keep, but then you say, but if it's absorbing something, isn't it pigmented?
Right.
But you sort of reserve pigments for chlorophyll, phytophyton,
Okay.
carotenoid pigments, psychobilly pigments. But the word pigmented means capable of absorbing.
Okay.
So instead we get around that by saying chromophoric. Chromophoric dissolved organic matter. But it means the same thing. Chromophoric means ability to absorb. We have problems with words. We have problems with vocabulary and jargon. And I say this not to be a pain, but just to get you to think about the words that you use and what your implication is. And the best thing to do is use the term you're going to use, but just define it in your papers. You know, I'm using this word. It's going to represent this for me. Okay. All right. So here's some absorption spectra. Oh, that is not the figure that needed to go in there. Oh, no, it's linked. Ah. Okay. All right. Anyway. Okay, I can't change it, but I can sketch it. I have it linked to an Excel spreadsheet.
Watch your wall.
Oh, no, I can't get on that side. right, I'm gonna draw it here.
You can erase our stuff, we have it written down.
You have it written down? So much work, you sure?
Yeah.
Oh, sure, okay. Yeah, I cleverly had this, it's linked to an Excel spreadsheet so that I could update things, but then I forgot something. All right, so we have, oh, excellent, draw slowly. This is wavelength, this is absorption. So what it should look like is that's one curve. That's the other curve. And that's the other curve. Estuary, coastal, open ocean. So if that were the case, what do they have in common?
They're very similar, after the 525 nanometer.
Yeah, so they all have this really strong absorption in this area here that's common to all of them, right? And so where are the biggest differences? At this end. And if you think about the environment of an estuary or a coastal ocean or the open ocean, what's the biggest difference in terms of what's in the water?
Sediment down.
Sediment, what else?
Down.
Sedum. And phytoplankton, right? And so what that tells you is that those materials tend to do a lot of their absorbing work in the blue and green. And not as much in the red. And for phytoplankton, you might think to yourself, they're pretty intentional about their pigments. Why don't they really have a lot of red absorbing?
Minimal red light.
Right, there's minimal red light because something else has taken it all out, which is water, right? So it wouldn't make sense. I mean, and they have that little red absorption peak, but that's the photosynthesis. We'll talk about that later in the class. Okay, so they all have red absorption. They differ in the blue, and so we tend to highlight most of our work in the blue. Okay, all right. So absorption's a conservative property. If you measure the total absorption, which you will with an instrument, it literally is the sum of all the individual molecules within all the individual particles or dissolved. So we could say it's water plus the sum of all the dissolved molecules and sum of all the particles. And in the particles, it's really the absorption of all the materials in the particles. So that's good. And the other thing about absorption is that it's proportional to the concentration, and that's Beer's law. And did you guys do any Beer's law yesterday? Not yesterday, okay, so yeah. I wasn't sure if there was one in this. You're doing Beer's law today. So Beer's law today says that the absorption is proportional to the concentration. So if I want to know the absorption of chlorophyll in water, it turns out that it's linearly related to the chlorophyll concentration multiplied by whatever the chlorophyll specific absorption is. So if I could measure the absorption of a molecule of chlorophyll, and I'd say, that's the absorption per molecule, or that's the absorption per mass. And that happened to be a constant. Wouldn't that be awesome? It is in some cases. Then the absorption is just going to scale with chlorophyll, because this will be a constant spectrum or something. Okay. So that's great. The more stuff you add to the water, the more absorption we get in a linear sense. So this math, or the fact that you can sum all of your absorptions to get the total, that it's conserved, and that each one of these things, each one of these materials is proportional to its concentration, makes absorption really easy. Great. Okay. So what we need to do is be able to measure the absorption by every constituent. And that's really hard. So instead what we do is we begin to group them into things that absorb similarly, because we can't go out. Well, it turns out one person could do that and measure the absorbed by every individual particle. I'll show you an example. Paper got published and no one ever picked up because it's really tedious, as you can imagine. But we know that we have water, and then we say CDOM. Well, there are a lot of different molecules in CDOM. They have, some absorb strongly, some absorb weakly, but let's just filter the water at 0.2, and anything less than that tends to have a really similar absorption spectrum. You can make a career out of the differences, but you can also be like, I'm a grouper. Okay? Let's group those together. So we'll filter it and we'll say everything smaller than that is CDOM. It all looks similar. And phytoplankton, well, phytoplankton are really different, but we'll just group those together because they're like real pigmenters. And then we have this term, NAP. I heard someone talking about NAP yesterday. Yeah. What is it? Non-algal particles. Everything that's not phytoplankton. So that's what we call detritus now. It's just not phytoplankton. So that is bacteria, viruses, if they get stuck on your filter. That is actual dead material. That is zooplankton. That are fecal pellets, aggregates, marine snow. All of that falls into NAP. If it's bigger than a filter and it's not a phytoplankton pigment, it gets put in there. Sediment can get put in there. And so you could add whatever else you want there. All right, so we group them by their common absorption properties and our ability to separate them. Yes, Emmanuel.
What about other material within phytoplankton that are not?
So here's the problem. Phytoplankton are more than just their pigments. They have cell walls, they've got membranes, they've got DNA. If you're able to take the pigments out of the phytoplankton and just have the rest of the cell, what do you think it's going to look like, and where should it go? Where? With the water? Is it particulate? Yeah, so it can't go with water. It's not water, even though it has a lot of water in it. Huh?
Non-algal particles.
Is it going to go in non-algal particles? Why not? Well, I know that, and you know that. But optically, it absorbs like non-algal particles. It's cellular material, just like the zooplankton cellular material. Okay, we'll get there, but excellent question, because this is where it starts to get sticky. So, yeah. All right, so let's talk about water. This is the absorption spectrum of water. You can see it has a lot of peaks associated with its quantized states. We are in the visible, we're here. So we're at the minimum of absorption. It absorbs really strongly in the UV. It absorbs very strongly in the infrared, and it's got all these really nice harmonics in its absorption. For such a small molecule, it really is astounding. I get astounded by it more all the time. So, all right, so if we put it on a linear scale here, this is what the absorption spectrum looks like, and you can see it's just like my sketch. But there are these little peaks, around 600 and around 670-ish. If you blow up the blue part, you can see those little peaks are in there too. Okay. This was from a very early paper, people trying to actually figure out what the absorption by pure water was, and they do a really good job here because the signal's so high, but as soon as you get into blue. So these are all like some of the original measurements. I think we have a pretty good handle on it now, but all those variations were methodological. All right, so the problem is when you measure the absorption at different temperatures, you get different absorption spectra. So what's happening here? Why is this happening?
That changed the original energetic state.
It's changing the vibrational levels, right? And so you're changing the probability of absorption as a function of temperature in the vibrational states, the quantized vibrational states. And so you get very, very big differences. And these are huge absorption coefficients, right? These are 2 point, and we're down here at like 0.2. Okay, so
Is it not also changing the rotational or just vibration?
yeah, so there will be rotational changes in there also, right? That's why it's nice and smooth. So this is way out, this is out in like 740. And we measure absorption in our AC meters and spectrophotometers out there. But you'll also notice that there's some differences here that we don't tend to notice, but they're there. There's some here too. So depending upon the absorption coefficient that you're trying to measure, if you're in the coastal waters, you might not be bothered with this. It's small relative to CDOM. But if you're out in the open ocean, these are very huge differences. So we have to take into account the temperature of the water that we're measuring. And we calibrate our instruments relative to water. And so we need to know the temperature of the water that we calibrated at. We need to know the temperature of the water that we're measuring, because the differences could be due to temperature. And you think, oh gosh, what a pain in the neck, until you say to yourself, I already paid my $40,000 for an ACS and I just got a temperature sensor, because I could actually use the variations in absorption here to estimate the temperature. So you just got a sensor you didn't buy, which is awesome.
Okay.
Yes. Right? So you calibrate your sensor at 20 degrees, and then you go and you measure in the ocean and it's 5 degrees, it's going to be lower absorption than your calibrated sensor. And you can quantify how much lower and get exactly what the temperature is. So if you're profiling, you could actually get a profile temperature. So
It's an expensive
Bonus.
temperature code.
every headache is an opportunity, right? If you walk away with anything from this class, I hope you walk away with like, oh no, my measurement depends on X. Instead, you'd be like, hey, I can measure X, right? Because if it's causing your measurements to vary, then that means you can use your measurements together. Yeah. Did you have a question? Okay. So part of the thing that's happening is that water molecules are clustering and they're bonding together with hydrogen bonds. And the size of those clusters varies with temperature. And so that's also going to sort of change some of the absorption coefficients based upon the clustering of this water. Also, and so you can calibrate absorption versus temperature, and then we use that to calibrate or to calculate our temperature effect of our water. Also, you can add salt. So as you add salt, you get changes in the absorption, because it's changing the molecular structure of how water molecules bond to each other, and they cluster around the dissociated salts. We're going to do that in the lab. And so you can see the variation in absorption of salt concentration. And this, you'll become very familiar with this. This is actually the correction for absorption based upon salt. And so there's your spectrum from blue to red. And so it makes a difference if you calibrate with fresh water. If you're then measuring the ocean, that's 33 parts per thousand. You'll have to correct for the absorption by the salt. So you could use it as a salinity sensor as well. So now you just got yourself a whole CTE. Further into the ultraviolet, we can see absorption features by hydrogen sulfide, by, let's see, that's hydrogen sulfide and Meg with HS. Yeah,
nitrate?
This one, I can't remember what HS is. Anyway, if someone wants to measure HS, you can get that. But yeah, here's nitrate, right? Who doesn't want to know how much nitrate's in the water? Well, it's this absorption right here. Sadly, it overlaps a lot with bromine, which is one of the major absorbers because of salinity. But it's a very different width of absorption. This one drops off very quickly, whereas the nitrate drops off slowly. So that has been exploited in a UV absorbing instrument to get this part, the magnitude of the nitrate absorption out here, and now you have a nitrate sensor based on absorption. That's called the ISIS.
I was hoping you could just quickly go over the absorption harmonics you mentioned with water.
This one.
No, it was a couple before. Yeah, those harmonics, like
Yeah.
what those are.
So those are different vibrational states.
And so-
They're vibrational harmonics.
Okay.
So depending upon the vibrational energy of the molecule, the magnitude of each of those harmonics will be different. And so when I showed you the difference between 5-degree and 30-degree water, that was a vibrational harmonic.
Okay, so it seems to me there's really not that many of them compared to what you might expect. I mean, that's sort of-
So there's a lot of them in the infrared,
Okay, so they process over less time.
and this is a log scale. If you blow up the visible right here,
So in these more defined peaks, are there certain vibrational and rotational transitions
Yes.
that are much more probable than all the rest?
yes, exactly. And so when we get to here, now we're in the part where we have minimum absorption. The harmonics are smaller, but for us, those can be significant. Does that make sense?
Yeah.
Yeah. Okay. So now we have a CTD, a nitrate sensor, all with an absorption here. Okay. So for the components, the definitions came about. It's because of how we handle our samples. So if you take a water sample, in an ACS, you can measure the whole thing. You just measure the water sample. You can measure it in a spectrophotometer on the benchtop, but their path length isn't long enough, and we often can't get that measurement as well. So you can separate the particulate and dissolve by filtration, and it's all dependent upon what filter you're using. And so how many people measure CDOM in their work? Anyone? What filter size do you use? 0.7. 0.2. 0.2. Okay. I'm sure you guys can find commonality in your data sets. What happens between 0.2 and 0.7? Yeah, colloids. Right? I actually have a whole data set that I've collected for my whole career. Everywhere I went, I always do 0.2 and 0.7. Why? Because the manual always does 0.2. Right? So I have to have 0.2 so that Emmanuel and I can make sense of our data together. But I use 0.7 also because that's how I get my phytoplankton on a glass fiber filter. The GFF is 0.7. Right? So I have to have that. So I have closure of phytoplankton, my phytoplankton, and then everything else that passes through. So I have that data set. And then I have the data set of 0.2 to 0.7. So your filter matters. You want to make sure that you have closure on your filtration. Okay. So if we measure the absorption by our dissolve fraction, we get some exponential. We'll talk about the exponential. But that's with the absorption. It's increasing into the UV. It's negligible in the infrared. And then we have all of our particulate matter, which I've colored green because it's, in my mind, the phytoplankton is what I'm interested in. It could be brown, depending on where you are. So here's some examples of the CDOM. This was from the book by John Kirk, where he was measuring CDOM in a variety of freshwater lakes in Australia, where the absorption coefficient is huge, very similar to our lakes here. Here's a beautiful photo from Heidi Dearson, where she was out paddling with her white paddle. The water is just so brown, almost red. And then, of course, the beautiful Clark Little CDOM wave. Okay. So because it has this nice smooth shape, people want to come up with an analytic function for it. So they define it using an exponential function. People have tried to use different types of analytic functions, but this one keeps seeming to win out. What it says is, the reason why you do it is it says that basically, if you know the steepness of this slope, and if the steepness of that slope was constant, you really need to only measure absorption at one wavelength. If you measure it at a reference wavelength, and this number is a constant, you can reconstruct the entire spectrum. And so it becomes incredibly powerful. In reality, this number is not constant, and we can always measure this one. But if we know something about this, it can tell us something about the material. And so Ken Carter first published this back in '89, where he was looking at the currents around Florida in the loop current, and he looked at different chemical extractions of fulvic material and humic material, and found that if you take this spectrum and you put it on a log scale, you take the exponential shape and you get a line. The slope of that line is that exponential slope. And you can see that the slope of this line varies, and that humic material tends to be flatter and fulvic material tends to be steeper. And he said, so if I go out and I measure the spectral slope, I can figure out compositionally, in a qualitative sense, what material is in my white sample. Is it coming from the ocean or is it coming from the Mississippi River? So you could use that spectral slope as a tracer. Yes.
Is humic from land or sea?
So there's marine humic. There's also like inland water humic.
Okay.
Right? And so in his system, and I'm going to actually defer to Meg on this because she actually did work down there in Atchafalaya. In his system, the loop current had more marine-derived humic material, detrital breakdown products from marine ecosystems, and then the signal coming from the Mississippi River was more highly metabolized, refractory fulvic material, tannins, things like that. And it has a steeper slope. And so what I notice in the samples that I work on is when I have material that is more refractory, meaning less biologically yummy, that material tends to have a steeper absorption slope than freshly degraded phytoplankton bloom dissolved organic matter. So when you ask me whether the difference between detritus and sedum, and I said it's continuum, you also have this continuum into the sedum based upon the breakdown of the material, which of course means if organisms are metabolizing the organic molecules and then making them, they're removing the parts that they're eating, the nitrogen, what they need, they're changing the molecular structure. It turns out that in a general sense, the material that results from all of that metabolic breakdown tends to have a steeper absorption slope, tends to be more refractory, and tends to stick around. And so one of the studies that one of my students did, we're not there yet. Yes, so this is it. So that's the spectral slope of 0.01, that's the spectral slope of 0.02, log absorption versus wavelength. That would be what would go in your exponential equation. I had a student that did the 0.7 to 0.4 0.2 to 0.7 comparison. And she said, okay, this was in the equatorial Pacific, along the equator. And what she found was that as a function of depth, when she looked at the fraction of sedum that was less than 0.2, the spectral slope was 0.04, which is much steeper than the spectral slope of the fraction that was 0.2 to 0.7. If we follow Ken Carter's work, we would say this size interval, 0.2 to 0.7, was more labile, was more yummy, was more freshly dead than the smaller fraction. And if that's true, then when we look at the distribution, what we found is that there was more of that material down deep. This was an upwelling event of deep water coming up. It was deeper, it was probably older, it was probably had more time to be degraded. This one had higher concentrations in the surface and was associated with two distributions of phytoplankton. Fresh, less broken down, more labile. So then you say to yourself, well, A, measure 0.2 and 0.7. B, look at the spectral slope. Use that spectral slope information to tell you something qualitatively about that material. And so you can use it as a tracer for deep water material, deep water soils. Okay, so, this is just looking at the slopes of CDOM and how they vary for all of these different studies. This was actually from my PhD thesis when nobody was measuring CDOM except these people. And so I digitized all their data and calculated the slopes. And what you see is that the slopes are relatively similar. And they're on the order of 0.04 to 0.02, 0.014 to 0.02 in terms of steepness. Later, I did the detritus, because that's what we called it back then. And you see that they have an exponential slope that's much flatter. So the fresh detrital material has a spectral slope that's flatter than the dissolved. Okay. Marcel Babin did a huge survey of European coastal waters of the spectral slope of CDOM, and he found that his mean and his spread were exactly what I found for the globe. So it's pretty robust. CDOM does not have a huge range in slopes. Yeah, exactly. Okay, so then you take these particles and you measure them in a spectrophotometer. And this is from a paper by Kishino in 1985, the first paper that was able to separate phytoplankton from detritus, but we would now call it non-algal particles. They measure the spectrum. Then they extracted a methanol, which removes the pigments, leaves behind all the other stuff on the filter, including phytoplankton cells, the phytoplankton membranes, cell walls, nucleus, DNA material. They scan it again, and you get this scan. What's the shape of that remind you of? CDOM. But this is particulate. Then, if you subtract that from this, by difference, you get what? It should be the pigments, right? Not just chlorophyll, but all the pigments. But most importantly, by doing it this way, you're getting the absorption by the pigments as they were packaged in the cell. Because you could also measure the extract, but that's going to have a very different absorption spectrum because now the molecules are not attached to each other or anything, and they're insolvent. So this technique allowed the reconstruction of absorption by phytoplankton pigments in vivo, in a living cell. And it turns out if you do it in a culture, your non-algal particle absorption is very small. If you're good at growing cultures, there shouldn't be a lot of excess material in there. If you do it out in the ocean, you can get varying amounts. And if we compare that to someone who actually measured the absorption on an individual cell, it's called microphotometry. It's basically a spectrophotometer in a microscope. And Rodolfo Turiaga published this work. These are absorption scans of individual cells. And if you add them all up, it's like what you measure on a filter. But it's a ton of work. and no one wants to do it anymore. But what it showed us is that you could actually get absorption by individual cells. Okay. The variability in the absorption spectra, there's these are different species of different cultures. And you can see that everyone has the red chlorophyll peak and the blue chlorophyll peak, and all these other peaks are all the accessory pigments. You can identify them, the carotenoids, fucoxanthin, chlorophyll's A, B, and C. You get good at looking at shoulders and knowing exactly who it is. Phycobilly pigments, which we find in cyanobacteria and some of the other eukaryotic species, phycourobilin, phycoerythrin, phycocyanin, they tend to absorb more in this orange region here. I will tell you that these are not extractable with methanol, so when you do that technique, these get left behind. So you have to extract them with a hot phosphate buffer, boiling water. Works, gentle treatment with boiling water. And so when you do this method and you look at the absorption, you can see on samples very big differences in the shape of the absorption. And here it's been normalized to the blue peak here. And you see it's from the Neeper-Coe's work. Really, really different shapes in absorption of phytoplankton from different areas. And you can see that if you took your absorption and you normalized it to the chlorophyll concentration in your sample, we call that the chlorophyll specific absorption. There are some places of the world where you have really efficient absorption and some places where you have very inefficient absorption. And I think what I'm going to do, I was going to talk about absorption efficiency of phytoplankton, but I think I'm going to move through this part and talk about it later and give a more hearty treatment to pigment packaging, because I think it's going to come into play really importantly when we talk about why phytoplankton absorb light in the first place for photosynthesis. So I think it's important to recognize that there are very big differences in the absorption by phytoplankton, and then we'll talk a little bit more about why that is a little bit later. So I'm going to skip through some of those slides. Okay. The other organisms that absorb, these are other protists like ciliates and flagellates that don't have specific photosynthetic pigments or heterotrophic bacteria. What do you notice about their absorption spectra? Anything look familiar?
There's the blue peak, but not the red peak.
Yeah, so you have a blue peak but not the red peak. Good. But it turns out that this peak is at 412 and not at 440. That's actually a cytochrome. That's cytochrome 412 is involved in respiration. And when you culture bacteria and some of these other ciliates, you get a very strong absorption peak there that you don't necessarily see in the ocean. Sometimes you can see it in the ocean if you have a huge heterotrophic bacteria bloom, like say, the sewage runs out into the bay and people measure the absorption. You can actually quantify the bacteria with that peak. But the rest of it is this exponential, right? So it looks just like detritus, actual detritus, but it's living. So all of that material, the non-algal particle material, you can also fit an exponential curve to it, and you can look at its slope. And I found that the average slope was about 0.01, which was significantly lower than CDOM's slope. So that's good, at least you can separate it that way. Marcel found the same thing when he looked at the European coastal waters, almost identical value of the average spectral slope. So it's flatter. It's a little bit flatter slope. CDOM is more like this. Detritus is, non-algal particles is more like this. They measured microphotometry of detrital particles. Before they knew how to do the extraction, some people could photo bleach. You could take a culture and stick it under UV light, like a sun lamp. I don't know how many of your grandparents had sun lamps where they used to sit in front of them to get tan before they used to cause cancer. You can put them in front of that UV light, and you can actually degrade the pigments down and almost begin to reconstruct this non-algal particle absorption by removing pigments that way. And now we get into the inorganic particles, which show up in NAP. Here's the absorption by 3 different samples from work that Marcel and Darius did. Saharan dust, some samples from Spitsbergen in Australia. And you see, you know, you could fit an exponential to that. It wouldn't be perfect, but it shows up too, right? There's some other absorption peaks that, you know, you wouldn't want to interpret as a pigment. It's not, it's sediment. If you take the log of the absorption, you get a straight line, and the slope of that line is about 0.01, just like the organics. So Meg, in her thesis, did some really interesting work where she was measuring the absorption of samples from the Atchafalaya Mississippi rivers, and she measured the absorption of the particles. You did it in suspension, not on filters. So she measured the particles in suspension, then she measured the mass of those particles and tried to come up with, like, wouldn't it be great if the absorption per mass was constant, because then we could choose a single number. And so she got, you know, you could see that there's not a huge range, but she got this really nice mass-specific absorption spectra. They, of course, look very exponential. And then, but what she noticed, because she's a detail-oriented person, is that there are these little shoulders in there. Right? See those little shoulders? Get very used to shoulders. They're definitely sources of information. So she took the second derivative of the spectra and found that those result in this very strong peaks. And you could use the magnitude of the peaks in the second derivative, and it turns out it's related to the concentration of iron oxide. So again, taking something that causes variability and then using that to provide you with a method to estimate the concentration is something that's really difficult to measure. So if you want to talk to Meg more about that work, it was really excellent work. And so for modeling, if you want to model absorption, you can just measure all the constituents and add them up. Or by inversion, you can measure the total absorption and deconstruct it into various components. If we have phytoplankton, CDOM, NAP, and water, we can just add them all up. And we'll talk a bit later about this beautiful paper by Morel and Pierre about variations in absorption and reflectance. And you can see that this would be a very low amount of components. This has a lot of CDOM in it, and this has a lot of phytoplankton in it. And by looking at the shape of these curves, they call this the V shape, they call this the U shape, and they call this very water-like. But by looking at that shape, it kind of tells you who's in water. All right. So now you know why absorption has peaks, why those peaks are broad. You know how it varies based on the composition. You know who's in the water. You know how they absorb. You know why we group them together, even though we know it's not right. Why we call them certain names, even though we know that's not correct, but you can do it correctly. And we're going to learn how to make measurements in a little bit. Questions on this? Think about your questions. We have sticky notes. I brought bigger sticky notes today for bigger questions. I didn't get a chance to read your comments from yesterday, but I will. Sorry, my son had a baseball game and we still have house guests. So, but I will read them and get back to questions. And I would like to talk about those maybe before dinner if we have time today. I don't know. I'm very excited to see them. Was it fun?
It was a bonding experience.
That's actually the intention. So good bonding experience,
Yeah.
tedious exercise.
Yeah.
Yeah. So are you gonna love any classification scheme that someone comes up with that's automated?
Yes.
Yes. Will you have a good appreciation for how it distinguishes between phytoplankton?
Yes.
I don't know if this is yeah, great. So then the intention is to detail. That's exactly what I wanted you to learn. It's tough. But when we put these up and we can kind of lay them out by season, we'll be able to see some variations of how things do in season. And then I will tell you that because of the giant tidal range, you could do the same thing over a tide cycle. And you will see very, very different things at high tide than low tide out here. So, all right, take a break.